Hey everybody. We're here to talk about enzymes and metabolism and like types of reaction. If you're watching these all at once, you should know that my kids came out and were like, "Mom, you have your sweater on inside out." So I flipped it. So if you noticed that, which you probably didn't because there's not a tag in the front, know that the first 3 videos I am wearing everything inside out. So let's get into this right now. So let's first talk about some energy stuff. I would say this is the easier stuff. There we go. I think that's how we like it. The laws of thermodynamics. So the first idea is that we can't create nor destroy energy, but we can transfer it, right? We can pass it along, and every time it kind of like changes form, we're going to lose some in the form of heat. And that kind of brings us to the second law, which is that disorder or entropy is always increasing. That when we're changing, we're going from one form to another, we're losing some useful types of energy. And so this is where free energy came into play. Change in G is technically Gibbs free energy. Ooh, that equation is not going to be on the test. Again, this is an old presentation that I was able to dig up. I honestly think my newer one is at school on like a jump drive, which is weird. All right, so let's talk about types of reactions. This is like a big T-chart you could do. So we did exergonic versus endergonic. An exergonic reaction is one that releases energy. You guys called it exothermic, I think, when we talked about chemistry. So we're releasing free energy, which means that we've got a negative change in energy. You're going from the ability to do work here down to here. You're giving off energy, so your change in G is negative. We're talking about a catabolic reaction, which is one that's breaking something down, like glucose, breaking down a bigger molecule into smaller molecules and releasing energy, like ATP. It is spontaneous. Nope. Yep, it is spontaneous, which means it happens on its own. I feel like I'm forgetting something, like one of our little facts. An endergonic reaction is one that you have to like feed energy into continuously, because at the end, you're going to hold more ability to do work. You're making a product that's bigger, that's like going to hold more potential energy to do work. So that's an anabolic reaction. You're building a molecule. This is like photosynthesis. We're building glucose, right? Like we're putting in light energy to build glucose. So ATP in general is our energy currency that we're going to talk about. You guys are very familiar with this. Basically, the addition and the subtraction of the phosphate group right here are what's going to determine the energy potential. You don't really have to know a lot about it. So let's talk about catalysts. When we're talking about these reactions, especially our exergonic, you got to put a little bit of energy in, and then it will proceed on its own. So that little bit of energy is called the activation energy. You need to bring it to a state right here where it can then proceed on its own. Okay, so oftentimes that's supplied in the form of heat, like you're heating something up. A catalyst then is something that's going to speed this up but not be consumed. So the catalysts that we're going to talk about are enzymes. They're not always enzymes. They can be non-organic when you're talking about chemistry, but in biochemistry, they're enzymes. The idea is that we're going to lower the activation energy. The starting point and the finish line are the same in terms of net energy gain or loss, but the activation energy is less, and so it's going to speed the reaction up, okay? So this is kind of a classic graph. Oh, the words are all messed up. I'm sorry. The black line is an uncatalyzed reaction. You see reactants, you see products. Right here is the activation energy. And then when you add an enzyme, when you add a catalyst, it's going to start the same place, end at the same place, but it's going to take less to get going. And so you're lowering that activation energy by adding the enzyme. Okay, you should be able to recognize that graph is something I think you might actually see on an FRQ. It's a huge concept. And so let's talk about some basics with an enzyme. So the substrate is what we call the reactant. That's like the biochemistry way of saying reactant. The enzyme is the catalyst. The substrate binds to the enzyme. So there's this like lock-in key kind of thing where only certain substrates go with certain enzymes and vice versa. And then the active site is where that substrate binds. And so the shape of the active site is very important. The substrate has to be able to fit into the active site, and the shapes must be correct for the enzyme to do its job and catalyze the reaction. Okay, so here's our substrates. This was like my dollar bill demo. Oh my God, that would have been so fun to redo if I would have thought of that. I have a paper clip. I don't have any money. So these, in this case, there's two substrates. They land in the enzyme. The enzyme catalyzes the reaction. You don't really have to know how. And in this case, we were making two products. The products are released. The enzyme does not get consumed. It can be used again. And so we did like a fair amount of lab research when it came to enzymes and stuff. We did the catalysis lab, like the potato, the discs floating, like that whole thing, converting hydrogen peroxide to water and bubbles. Okay, so enzymes have certain conditions that they function best in. One of them is temperature. So you guys know, and like again, I think this is something the FRQ could talk about. So when you talk about temperature, in general, when you heat things up, reactions speed up. Okay, because the substrate is hitting the enzyme more frequently, right? Like the kinetic energy is increasing, but at some point it gets too hot, which you're going to see here. The enzyme denatures. That's that word that you should know. And the whole thing just like falls apart. So denaturing means the shape changes, which means the active site is not the same shape, which means the substrate can't bind and the enzyme can't catalyze the reaction. You should be familiar with that. Another one is pH. So enzymes have an optimal pH in which they can function. If it's too acidic or too basic, the enzyme will also denature. It will fall apart and the reaction cannot proceed. Okay? This is not a reversible thing either. Salinity might be on there. Salinity doesn't usually affect an enzyme. Temperature, pH. Substrate concentration. So substrate, as you increase more substrate, again, more product's going to be made. Unless you've like maximized active sites and then you kind of just level off. If you increase amount of enzyme, that would obviously increase the reaction rate until it like eats up all the substrate. So like think about all those factors that we tested way back when with the enzyme lab that you could be asked about. Because again, these FRQs are going to look at like research design and analyzing data and interpreting graphs and like stuff like that. Good. So when you're talking about inhibition, that's like another way that an enzyme could be stopped. So we talked about two types. Competitive inhibition is where the inhibitor is going to land right into the active site. So the substrate can't fit. So the enzyme can't be catalyzed, okay? Non-competitive inhibition refers to an allosteric site, if you remember that word, which is remote from the active site. The enzyme can no longer fit because when the non-competitive inhibitor binds, it changes the shape of the active site. Okay, so that should be it. Oh no, one more thing. Oh, so important. Feedback loops. So feedback inhibition is basically a negative feedback loop. So we have our substrate, it lands, there's a series of metabolic reactions, which is usually the case, like a metabolic pathway. It's not usually just one. And then we're going to make something. And when we have enough of this thing, it's going to go back and bind, and it's going to shut off the reaction. It's going to inhibit it so that it can't do it anymore. This is like when we talked about operons. Like this would have to do with operons. Like when you're making tryptophan, all of a sudden you eat it, it like shuts off the mechanism that makes it. Okay, so this is a great example of a negative feedback loop. And again, we're trying to save ATP. So why should that enzyme be catalyzing the reaction when you don't need to make any more of this end product, this isoleucine? All right, good. Come back and join me for cellular respiration in a bit. Maybe get a snack.